I am Emanuele Plini, fully certified clinical neuropsychologist, currently pursuing a PhD in neuropsychology and neuroimaging at Trinity College, Dublin, Institute of Neuroscience. I am supervised by Professor Paul Michael Dockery and supported by a scholarship provided by the Irish Research Council. I'm going to present a research I carried out together with the help of Max Planck Institute concerning the involvement of the tyrosine intake, the volume of the locus coeruleus, and the intercorrelation with attentional measures. So, very brief introduction. We know that dementia can be only prevented and that diet is a crucial factor for preventing strategies. We know also that protein intake is crucial for health maintenance, particularly for brain and cognitive health. There are several studies, possibly because in proteins there are amino acids which are precursor of our neurotransmitters and neuromodulators. In particular, tyrosine is the precursor of dopamine, and from dopamine we obtain norepinephrine. Now, a very brief overview of the tyrosine literature. We know that the ingestion of tyrosine can increase dopamine and noradrenaline in the blood, and several studies throughout the decades found that tyrosine administration in acute were related to better cognitive performances, particularly working memory and attentional tasks. There was also an interest of the US Army where tyrosine was administered in cold exposure in order to restore the detrimental effect of cold exposure. But anyway, the current consensus is that tyrosine intake may be beneficial for cognitive functioning, particularly in stressful situations. Very interestingly, a few years ago, people from Max Planck Institute, colleague, found that the habitual tyrosine intake was related to greater fluid intelligence, greater working memory, and more accurate episodic memory across the Berlin aging study too. So going over the aim of my study, I wrote to them and I asked if I could use that dataset to investigate whether tyrosine intake was related to greater locus coeruleus volume. Why? Because a few studies found that choline intake, which is the precursor of acetylcholine, was related to better cognitive performances and better brain health. So, performing analogical reasoning, I thought that maybe tyrosine intake could be related also to neuropsychological performances, but possibly even to the volume of the locus coeruleus, which is a key structure heavily affecting neurodegenerative diseases. So, going over the methodology, I proposed to investigate whether yearly tyrosine intake was related to the volume of the locus coeruleus, and also if tyrosine intake was related to better attentional performances, and overall if there were intercorrelation between these variables. So going over the actual methodology of EMRI analysis, I performed voxel-based morphometry analysis implementing multiple regression models. So the volume of the locus coeruleus was related to the yearly tyrosine intake, covering for age, total intracranial volume, gender, and education, all treated as continuous variable. The same analyses were repeated for the other main neuromodulatory seeds, like the dorsal raphe, median raphe, the ventral tegmental area, and the nucleus basalis of Maynard. This in order to better differentiate a possible relationship between locus coeruleus and tyrosine, and also to have a better idea of these results. Going over the results, greater tyrosine intake was related to greater volume of the locus coeruleus. The results in the locus coeruleus overlap the previously published LC atlases and masks. This is the relationship: greater volume of the locus coeruleus related to greater tyrosine intake, average habitual tyrosine intake. We can also observe that the blue region has a pretty high Bayesian factors, which is 8,000. It's very, very high, extremely high. And the more lighter region, which are 28 voxels, has an extremely high Bayes factor of 65,000. So I will say that this relationship is pretty strong, and these are the screenshots from JAST, which is a freely available statistical tool. The relationship between LC volume and tyrosine intake is the same in old and young people. Also, given that I performed contra-analysis, I found also that 11 voxels and 22 voxels in the dorsal raphe and the ventral tegmental area were related to greater tyrosine intake. There is not much time to explain this, but I believe that makes sense because tyrosine is the precursor of dopamine as well, and that dorsal raphe also has a noradrenergic component. But if we observe the Bayesian sequential analysis, we can see that the relationship between LC is more linear. So briefly going over the conclusions, I think that these results might help us to better understand the role of protein intake, particularly concerning certain amino acids like tyrosine, for brain health and cognitive maintenance, particularly in the phase of neurodegeneration, because we know that Locus Coeruleus integrity is crucial for the development of Alzheimer's disease, of Parkinson's disease, and these might be also help us to explain reasons why dietary lifestyles are crucial for preventing neurodegenerative diseases.